Building miniature brains may sound like a page out of a science fiction novel, but fact is indeed stranger than fiction. Researchers around the world grow brain organoids, 3D miniature brains, to better understand brain development, aging, injury, and other disorders, as well as to test new treatment strategies. Some scientists take brain building to the next level by launching their brain organoids into outer space. Welcome to The Scientist Speaks, a podcast produced by the Scientist Creative Services team. Our podcast is by scientists and for scientists. Once a month, we bring you the stories behind newsworthy molecular biology research. This episode is brought to you by Acro Biosystems and Molecular Devices. Acro Biosystems is a cornerstone enterprise of the pharmaceutical and biotechnology industries. Their mission is to help overcome challenges with innovative tools and solutions from discovery to the clinic. They deliver solutions, whether they come through recombinant proteins, antibodies, assay kits, GMP-grade reagents, as well as organoids, to be used in discovery research and scalable to the clinical phase and beyond. Molecular Devices is one of the world's leading providers of high-performance bioanalytical measurement systems, software, and consumables for life science research and pharmaceutical and biotherapeutic development. Included within a broad product portfolio are platforms for AI-driven automated cell culture workflows, high-throughput screening, genomic and cellular analysis, colony selection, and microplate detection. These leading-edge products enable scientists to improve productivity and effectiveness, ultimately accelerating research and the discovery of new therapeutics. In this episode, narrated by Nikki Spahich, Iris Kulbatski from The Scientist spoke with Alysson Muotri, a University of California, San Diego professor and stem cell program director, to learn more about how microgravity affects the cellular and molecular biology of brain organoids, and how these discoveries can improve human health.
Cells and tissues change in response to microgravity in outer space. Researchers have known this for many years by studying the bodies of astronauts after they return to Earth. Astronauts experience bone loss, muscle atrophy, anemia, central nervous system dysfunction, and other deleterious effects. Scientists study these phenomena by creating organoid models, shipping them off on NASA space missions, and studying the cellular and molecular changes that happen in a microgravity environment. They also use organoids that travel to outer space to model diseases when the organoids return to Earth. Alysson Muotri uses brain organoid models grown from human-induced pluripotent stem cells to study neurological conditions, such as those that occur during embryonic development or later in life.
Traditionally, neuroscience has been using mouse models or human postmortem tissues or blood, but all these models have limitations. Postmortem tissues are dead. You cannot do electrophysiology. You cannot study synapses. Mouse models, they evolve very differently from the human brain. And certain conditions are human-specific. If you're looking for problems in the brain, blood cells don't make synapses, they don't fire, they don't process information. I started working with human embryonic stem cells, then saw the Yamanaka revolution where we could reprogram cells from people. I started doing that for autism, comparing stem cells derived from autism to neurotypicals. Then in 2008, there is a revolutionary paper who shows us that if you start differentiating the stem cells in a three-dimensional fashion, they will self-organize and form what we call now a brain organoid. The cells will start migrating and forming a tissue that resemble, from the anatomical perspective, the developing human brain. I thought that's exactly the model that I want.
The cells that make up brain organoids mature in very similar ways to that of a living human embryo, recapitulating early neurodevelopment in a remarkably accurate way. Muotri's team has even been able to obtain recordings of electrical activity from the neuronal networks of brain organoids. Yet, regardless of how long they are kept alive in vitro, their growth and development plateaus for unknown reasons.
Maybe they're lacking input and output, or maybe we're missing other brain regions, or even contact with other parts of the body. I started looking for ways to age the brain cells inside the organoid, push the model towards a more developed human brain. I came across some early papers where investigators send cells to the International Space Station. The cells, upon return to Earth, show signs of accelerated aging. This has also been seen in tissues from the astronaut. I thought maybe I can leverage that for my experiment. I can push the developmental stage of these organoids a little bit further. We start working with bioengineers who help us to create autonomous platforms that can go into the rocket and be delivered to the space station.
This box, the Space Tango Cube Lab, is a miniature incubator and bioreactor that houses the cells and is equipped with computer hardware for automated control of cell maintenance and monitoring. The platform enables automated media changes, an agitation mechanism to improve growth, and a microscope for remote imaging. The logistics of sending brain organoids on space missions requires careful planning and execution. Despite this, there are often surprises and challenges that are beyond the control of Muotri's team, such as limited space on the rocket, infrequent takeoff dates, or last-minute weather-related cancellations. The pre-launch preparation of the organoids is extensive, with a 4-week-long advanced kickoff in the lab on Earth, when Muotri's team initiates the organoid cell cultures. Once they are mature enough, the brain organoids are ready for spaceflight. They are transferred to Cape Canaveral in Florida, where rocket launches to the International Space Station occur. When Muotri's team arrives at the Kennedy Center, they place the organoids into specially designed containers for their first-class travels to outer space.
These boxes with fully autonomous machines and microfluidic chambers keep these organoids alive by controlling temperature, humidity, CO2. There are even cameras installed inside these boxes so we can visualize the growth. We place these boxes inside the cargo, which is the Dragon module inside the SpaceX rocket. The battery is running the system, so it keeps everything alive. From the rocket to the space station usually takes about 2 days. Once it arrives, the astronaut has to plug the box inside one of the cabinets that has an outlet, so it gives power to these boxes. We also have access to the inside of the machine. Our missions last about 30 days. They arrive there when they are about 4 weeks, and we keep them over there for another 4 weeks in a microgravity environment. After the end of the mission, the astronaut removed that boxes from the outlet, placed back into the Dragon module. That would be splashed down here on Earth, somewhere in the ocean, where we have a boat that will go and rescue that. That might take another 2 or 3 days. That boxes is delivered back to our labs where we can rescue the organoids and perform further experiments. That's the journey of the organoids through space and back. We can fix the cells when they are in space, or we can bring them back alive. It really depends on the experimental design and the questions that we are pursuing.
Each brain organoid mission has both an element of repetition and builds on results from previous missions. In doing so, Muotri and his team work towards validating results and enabling new discoveries for space missions that occur infrequently. For Muotri's first ever mission, his team grew brain organoids from human stem cells. They found that the neural progenitor cells proliferated faster while exposed to the microgravity environment of outer space. But upon return to Earth, they aged faster, showing telomere changes and gene expression signs of cellular senescence, such as inflammation. Muotri is interested in understanding such mechanisms of accelerated aging and cellular time travel.
30 days in space would be equivalent of several years here on Earth. And we can measure that using several molecular tools, from gene expression to methylation analysis, pointing that whatever mechanism is happening inside the cells back here on Earth, they do suffer this speed up aging. And we think that this is related to the way the telomerase acts on the cells. Telomerase is those enzymes that controls the size of your chromosomes during aging. The telomerase activity seems to be paused in space, but as you bring them back to Earth, we see an overcompensation of the time that it has not worked. But we see other molecular pathways, for example, reactivation of endogenous virus in the genome. Our genome contains a lot of these endogenous retroviruses that are left over from human evolution. They tend to be very silent here on Earth. But when you send the cells to space, we see a reactivation of these retro elements. The brains, the cortex of Alzheimer's postmortem tissues are full of these retro elements. I think these are just molecular markers that are pointing us to the fact that these cells are aging.
Muotri's brain organoid work serves a dual purpose. It helps NASA understand how spaceflight affects the human brain and how adverse brain changes, such as lingering cognitive deficits, depression, and anxiety following long spaceflights, can be addressed. Post-spaceflight brain changes can often be severe and impact quality of life significantly. Muotri's work also addresses the need for robust, disease-specific models and research on healthy aging. Muotri's team uses brain organoids that travel to outer space to model late-onset diseases, such as Alzheimer's disease, dementia, and ALS. Good models for these diseases are scant because researchers aren't able to wait 6 or 7 decades to track the development of disease-specific phenotypes in humans. Exposing brain organoids to the microgravity environment of space allows Muotri to observe Alzheimer's-specific phenotypes faster and accurately model the disease. Previous space missions included organoids obtained from the cells of healthy individuals without late-onset diseases. But Muotri's team recently grew and launched brain organoids from cells obtained from Alzheimer's and ALS patients into outer space.
The organoids are, as we speak, still there. We haven't collected them yet. Our hope is that once they are back, we will reveal some phenotypes that we couldn't see on Earth. We are looking, for example, for Alzheimer's, defect on synaptogenesis, loss of synapses. These are the kind of defects that we see on the clinical setting, but very hard to reproduce in vitro. We hope that by aging the organoids, we will see a lack of synapses on the Alzheimer's brain organoids. And then the next stage would be, how can we prevent that? We are working with collaborators from the University of Amazon, where they have curated a very nice biodiversity library of molecules that seems to be neuroprotective. We're going to be testing these novel molecules coming from the Amazon plants to see if they are protective against Alzheimer's. Omics, global gene expression, single-cell gene expression, single-cell proteomics are really helping us to dissect what's happening in different cell populations. The organoid model is really cool because it has several cell populations. It's not a homogeneous population of cells, so we can start seeing the interactions. We are also building on other tools that neuroscience have developed for different purposes. For example, all optical electrophysiology we are taking to space as well. It's very hard to do patch clumping in space to record from a neuron because that depends on gravity. But we can do that with imaging. All the optogenetics, all the different reporters that we can actually visualize with a fluorescent microscope is very handy for us to look into the activity of those neurons.
Neuronal activity is critical to creating reliable and accurate brain models of disease. Yet as brain organoid research becomes increasingly sophisticated, scientists are beginning to grapple with ethical questions. The electrical oscillations recorded from brain organoids are similar in some ways to those of the human brain. As Muotri's team continues to optimize the protocols for growing brain organoids, the neurons that make up these miniature brains begin to form synapses, transmit electrical activity, and organize into more sophisticated networks.
As we start showing that it functions like the human brain, it makes some people uncomfortable. Our most recent work is showing that they have also plasticity, so they might be able to produce highly sophisticated tasks such as learning and memory. I think it's inevitable that the organoid technology will reach to that level where these brain organoids might develop some kind of a consciousness that might be similar to a lower animal. And at that stage, I think we might need ethical supervision in the same way that we use for animal models, or approval from an ethical committee. But that might influence if you're getting these cells from alive individuals. How do they feel about having their avatars or their clones in the lab that might reach a level of consciousness? Some people might decide not to donate their cells. These might affect how the science is progress.
Muotri envisions a future where his work generates increasingly sophisticated brain organoid models of neurological disease that approximate more closely the reality of the human brain. He also has his sights set on even loftier goals, sending scientists, not just brain organoids, on missions to outer space.
I'm very excited with the space results that we are generating. The experiments that we want to do now is becoming highly complex. I no longer can do it with a robotic microfluidic system. I have to do it with a human hands. My idea now is to make our scientists astronauts and to create a manufacturing facility in space where we can produce organoids and other tissues so we can speed up drug development here on Earth. What excites me about the future is creating a stem cell lab in space.
Thank you for listening to The Scientist Speaks. This episode was produced by the Creative Services team for The Scientist. Thanks again to Acro Biosystems and Molecular Devices for sponsoring this episode. Please join us again in July as we learn about how researchers use next-generation sequencing to elucidate rare diseases. To keep up to date with this podcast, follow The Scientist on Facebook and Twitter, and subscribe wherever you get your podcasts.